Imagine a vast library filled with ancient scrolls. These scrolls hold the instructions for building and maintaining every living organism on Earth. This is not a scene from a fantasy novel. This is the reality of DNA, the molecule that governs life. DNA, short for deoxyribonucleic acid, is the blueprint of life. It resides within the nucleus of almost every cell in our bodies, a microscopic library holding the secrets of our existence. Like a long, twisted ladder, DNA is composed of two strands of nucleotides linked together by chemical bonds. These nucleotides are the building blocks of DNA, each containing a sugar molecule, a phosphate group, and one of four nitrogenous bases: adenine, A, thymine, T, guanine, G, and cytosine, C. The sequence of these bases forms the genetic code, a language that dictates the production of proteins, the workhorses of our cells. The discovery of DNA structure in 1953 by James Watson and Francis Crick revolutionized biology. It unveiled the elegant simplicity of life's code, a code that determines everything from the color of our eyes to our susceptibility to certain diseases. The understanding of DNA's structure and function has opened up unprecedented avenues in medicine, agriculture, and biotechnology, promising a future shaped by our growing knowledge of this remarkable molecule. Within the sprawling tapestry of DNA, genes are the individual threads, each carrying a specific set of instructions. A gene is a segment of DNA that codes for a particular protein or functional RNA molecule. Like distinct words in a sentence, genes dictate the synthesis of proteins, which carry out a myriad of functions in our bodies, from building tissues to regulating metabolism. Genes act as blueprints for proteins, dictating the precise sequence of amino acids, the building blocks of proteins. The order of these amino acids determines the protein's three-dimensional structure and function. Just as a slight change in a word can alter the meaning of a sentence, a single mutation in a gene can have profound effects on an organism's traits and health. For instance, the gene for insulin, a hormone that regulates blood sugar levels, provides the instructions for cells to produce the insulin protein. Mutations in this gene can lead to the development of diabetes, a condition characterized by high blood sugar. The study of genes, known as genetics, is crucial for understanding the inheritance of traits, the development of diseases, and the vast diversity of life on Earth. One of the most remarkable aspects of DNA is its ability to replicate itself with astonishing fidelity, ensuring the transmission of genetic information from one generation to the next. This process, known as inheritance, is the cornerstone of life's continuity. During cell division, each DNA molecule creates an exact copy of itself, passing on its genetic code to daughter cells. This faithful replication of DNA ensures that offspring inherit traits from their parents. The genes we receive from our parents determine our physical characteristics, such as eye color and height, as well as our predisposition to certain diseases. Through inheritance, DNA bridges generations, weaving a tapestry of life that connects us to our ancestors and future generations. However, inheritance is not always a perfect process. Mutations or changes in the DNA sequence can occur spontaneously or due to environmental factors. These mutations, though often harmless, can sometimes lead to genetic variations that influence an organism's traits, for better or worse. The gradual accumulation of genetic variations over time, coupled with the forces of natural selection, drives the process of evolution. Evolution is the grand narrative of life on Earth, the ongoing saga of adaptation and change that has shaped the biodiversity we see today. DNA, with its capacity for variation and inheritance, is the protagonist of this epic tale. Mutations introduce genetic diversity into populations. Some mutations may be detrimental, leading to diseases or reduced fitness. Others, however, may confer an advantage, making an organism better suited to its environment. These beneficial mutations are more likely to be passed on to future generations, gradually increasing in frequency within a population. This process of natural selection, where organisms with advantageous traits are more likely to survive and reproduce, acts as a sculptor, shaping the genetic makeup of populations over time. The intricate adaptations we observe in nature, from the camouflage of a chameleon to the sonar navigation of bats, are all testaments to the power of DNA and evolution. Section 5. Epigenetics, Beyond the Code. While DNA provides the blueprint for life, the environment plays a crucial role in shaping how this blueprint is interpreted. Epigenetics, a field that has gained significant traction in recent years, explores the mechanisms by which environmental factors can influence gene expression without altering the underlying DNA sequence. Imagine a musical score. The notes on the page represent the DNA sequence, the fundamental genetic information. However, the way the music is played, the tempo, the volume, the interpretation, can vary depending on the musician and the setting. Similarly, epigenetic modifications act like musical annotations on the DNA score, influencing how genes are expressed. These modifications, such as the addition or removal of chemical tags on DNA or its associated proteins, can switch genes on or off, effectively controlling the volume of their expression. Epigenetic changes can be influenced by a multitude of factors, including diet, stress, and exposure to toxins. Section 6. Nature and Nurture. The interplay between genetics and the environment, often referred to as the nature versus nurture debate, is central to understanding human development and disease. While our genes provide the basic blueprint, our environment, including our upbringing, experiences, and lifestyle choices, can significantly influence how this blueprint is expressed. Epigenetics provides a molecular framework for understanding how environmental factors can leave their mark on our genes. For example, studies have shown that early childhood experiences, such as stress or neglect, can alter the epigenetic landscape of the brain, potentially influencing an individual's susceptibility to mental health issues later in life. The understanding that our environment can influence gene expression has profound implications for public health and personalized medicine. By identifying environmental factors that contribute to disease, we can develop targeted interventions and lifestyle modifications to mitigate risks and improve health outcomes. Section 7. Genetic switches. Flipping the script. Epigenetic modifications act like switches, turning genes on or off in response to environmental cues. These switches play a crucial role in regulating gene expression, ensuring that genes are activated or silenced at the right time and in the right cells. Just as a light switch controls the flow of electricity, epigenetic modifications regulate the flow of genetic information. One of the most well-studied epigenetic mechanisms is DNA methylation, where a methyl group, CH3, is attached to a DNA base, typically cytosine. This methylation can block the binding of proteins that activate gene expression, effectively silencing the gene. Conversely, the removal of a methyl group can turn a gene on. Another important epigenetic mechanism is histone modification. Histones are proteins around which DNA is wrapped, forming a complex called chromatin. Modifications to these histones, such as the addition or removal of chemical groups, can alter the accessibility of DNA to the cellular machinery that reads and transcribes genes. Section 8. The environment's imprint. Our environment exerts a profound influence on our epigenome, the collection of all epigenetic modifications in our cells. From the air we breathe to the food we eat, our surroundings can leave lasting marks on our genes, influencing our health and well-being. For instance, exposure to toxins, such as cigarette smoke or air pollution, can alter DNA methylation patterns, increasing the risk of developing cancer and other diseases. On the other hand, a healthy diet rich in fruits and vegetables can promote beneficial epigenetic changes, protecting against chronic diseases. The understanding that our environment can shape our epigenome empowers us to make informed choices that promote health and longevity. By adopting a healthy lifestyle, we can influence our epigenome in positive ways, potentially reducing our risk of disease and improving our overall well-being. Section 9. Diet and genes. The adage "you are what you eat" takes on new meaning in the context of epigenetics. Our diet provides not only the building blocks for our bodies, but also a constant stream of signals that can influence gene expression. The nutrients we consume can act as epigenetic modifiers, turning genes on or off and influencing our health. For example, folate, a B vitamin found in leafy green vegetables, plays a crucial role in DNA methylation. Adequate folate intake during pregnancy is essential for proper fetal development and can influence the risk of birth defects. Similarly, compounds found in cruciferous vegetables such as broccoli and cauliflower have been shown to have cancer protective effects by altering epigenetic patterns. The field of nutrigenomics explores the intricate relationship between diet and genes, aiming to develop personalized dietary recommendations based on an individual's genetic makeup and epigenetic profile. By understanding how our genes interact with the food we consume, we can make informed dietary choices that optimize health and prevent disease. Section 10. Stress and its genetic echo. Stress, a ubiquitous experience in modern life, can leave its mark not only on our mental well-being but also on our genes. Chronic stress can trigger a cascade of physiological responses, leading to alterations in DNA methylation and histone modifications, particularly in genes involved in the stress response. Studies have shown that individuals who experience chronic stress, such as those caring for a loved one with a chronic illness, exhibit distinct epigenetic changes compared to their less stressed counterparts. These changes can influence a range of physiological processes, including immune function, inflammation, and even the aging process. The good news is that just as stress can negatively impact our epigenome, stress-reducing interventions such as mindfulness meditation and yoga have been shown to promote beneficial epigenetic changes, reversing some of the harmful effects of chronic stress. By managing stress effectively, we can protect not only our mental health but also our genes. Section 11. DNA, the symphony of life. DNA, the molecule of life, orchestrates the symphony of our existence. It carries the genetic code, the blueprint of life, passed down through generations, ensuring the continuity of species. Genes, the individual instruments of this orchestra, dictate the production of proteins, the workhorses of our cells, carrying out a myriad of functions that sustain life. Epigenetics, like the conductor of this grand orchestra, fine-tunes gene expression, ensuring that genes are played at the right time and in the right cells, responding to the environment's cues. This intricate interplay between genetics and the environment shapes our traits, influences our health, and ultimately defines who we are. Understanding DNA is not merely an academic pursuit. It is fundamental to unraveling the mysteries of life, from the inheritance of traits to the development of diseases. This knowledge empowers us to develop new medical treatments, improve crop yields, and address some of the world's most pressing challenges. Section 12. A future illuminated by DNA. The unraveling of DNA secrets has ushered in a new era in biology and medicine. Advances in DNA sequencing technology have made it possible to decipher the human genome, the complete set of our genetic instructions, at an unprecedented pace and reduced cost. This knowledge is paving the way for personalized medicine, where treatments are tailored to an individual's genetic makeup. Gene therapy, a revolutionary approach that aims to treat diseases by correcting faulty genes, is showing promise for a range of genetic disorders. CRISPR-Cas9, a groundbreaking gene editing tool, allows scientists to precisely alter DNA sequences, offering hope for curing previously untreatable diseases. As we delve deeper into the intricacies of DNA, we are constantly amazed by its complexity and elegance. The more we learn about this remarkable molecule, the more we realize how much more there is to discover. The future of medicine, agriculture, and biotechnology is intricately intertwined with our understanding of DNA. The journey into the heart of life's blueprint has just begun.